All right, so this, I know if you're new to programming, you've seen like so many new things today. You may be feeling a little overwhelmed. But what we're going to wrap up with today is a topic that I think is really fun. So hopefully this part isn't too overwhelming, but we're going to be looking at this part. I'm hoping won't be. I'm hoping this part is fun. We're going to be looking at how we can visualize PDB files in the Jupyter Notebook. So this is a protein visualization portion. So we've been pulling information out of those PDB files. The PDB files are text files that give information about proteins. You can also visualize the protein based on the information in the PDB file. There are other softwares that exist that you can do this with, but we're going to use one that works in the Jupyter Notebook. So some information that's in the PDB file is like the location of all the atoms, like the coordinates, and we're going to visualize that location as well as some other information about the proteins. So if you go to your IQB.get tab, I want you to open the NGLView visualization student.ipynb. So just click on that. And you will see a notebook that looks like this. So this says you can use Python and the Jupyter Notebook to interactively visualize PDB files. We're going to be using another Python library called NGLView. I believe you have to be in the Jupyter Notebook to use NGLView. So for most of the other libraries that we've learned, there are other ways you can use Python, and you can use those other libraries in like text editors or other places that you use Python. But NGLView, I think, can only be used in the Jupyter Notebook. So it says, in this notebook, we will use the Python library NGLView to visualize data from PDB files. And so we're going to need to open a file, and we're going to need to use a function in NGLView. So we're going to import both of those libraries in this cell. So first I'm going to import OS, and I'm just importing OS because I know I'm going to be reading the file. And then I'm going to press enter, and then I'm going to import my protein visualization library called NGLView. And I'll do import NGLView. That's the name of the library. And then I'm going to type, so every time I use a library in Python, I type the library name and then a dot and then the function name that I want. NGLView is kind of a long library name, so Python will actually let us shorten it. So we're going to say import NGLView as NV. And what this means is that every time we want to use a function in NGLView, we're going to type the word, we're going to type NV instead of NGLView. So it's just a nice way to kind of shorten what we have to type. It makes it so we don't have to work as hard. Okay, so I'm going to type my import OS and then import NGLView as NV, and then I'll press Shift and Enter to execute. Okay, so now we want to read in a PDB file. So we need to build our file path. So I'm going to say file path equals, and then I'll do os.path.join, and we will say look in the PDB files folder, and then I'm going to put a comma, and I'm going to look at the PDB file fgu.pdb. So that is building my file path. And then I'm going to use the NGLView library to create a visualization of my PDB file. So I'm going to create a variable called view and set that equal to nv.showFile. So the name of the function that we're going to be using in NGLView is show file. And then I'm going to give the name of the file. And I just made that name using OS path join. Okay, so I am going to type that, then I'll press Shift and Enter. If I have constructed my file path correctly and Python found the file, this should execute without any error, and I won't see any output here. If we actually want to see our PDB representation, what we have to do is put this variable that we created called view in a cell, and it has to be the last thing in the cell. So if we want to see what this looks like, we just type the word view, and then we press Shift and Enter, and we should have a nice protein visualization here. So here it is. You can see that it's interactive. We can hover over it. I can grab it and drag and look at the protein. If you use your scroll, if you click on it and scroll, you can zoom in. You can see that we're getting different information here. So let's do a status check. If you were able to load your protein visualization, use the green check mark. And if it did not work for you, use the red X. And I'm curious if the people it's not working for are on Anaconda or not using KimCompute. It's a little, so the nice thing about KimCompute is that it's all set up to work for us correctly. This is using something in, I said this only works in Jupyter. And sometimes computers, sometimes you have to do extra setup if you're installing it locally to make it work. What are some things? Oh, I can click, there's a little button in the corner that will make it full screen. So that's kind of cool.
Also, if you go with the lower right-hand corner, you can drag it to make the window actually a bit bigger. Yeah, like that.
Yep. So this is very nice, very interactive. So if you want to, and we'll also, you also see that NGLView makes some really nice kind of default representations for us. So it's making the protein, it's using a representation called, it's using a representation called the cartoon and for the protein, and we'll talk about these different types of representations in just a moment. And then it's using something called ball and stick for the, this is a ligand, or two ligands, I think. But we can also change the representations. We can also change the representation, so we can change how this looks, and we can pretty much customize it to be anything that we want, and we'll see that in just a moment. I meant to, when I started, to tell you a little bit more about how the Jupyter Notebook works based on a question that I got. So someone was asking, I want to demonstrate this. So this is just a Jupyter Notebook thing that I'm showing you. I'm going off script a little bit. Someone asked if I forgot a command, how can I figure out maybe that command? So in the Jupyter Notebook, if I start and I type NV dot, and I think, I can't quite remember the name of that function. If I press tab twice, it's going to pull up a something that looks like this. This is a list of all of the functions that are available underneath that NGLView library. So you could look at this and you could be like, what is it? And here we'll see show file. So I could click on that and it would sort of type it for me. So that's something that's a Jupyter Notebook thing. If you forget a function name, you can start typing it and then press tab twice and it will show you a list of options that fit what you typed. Another thing to know, and this is also a Jupyter Notebook thing, is if you type a question mark at the end of that, it will pull up some information about like how to use the function. So that is something to know, and I did want to tell everyone about that. So that's just a quick aside on using the Jupyter Notebook that you might find useful. Okay, so we already kind of went over this. It says, you'll notice that NGLView represents the ligand and protein in different ways. And by default, it shows the protein in something called a cartoon representation and the ligand in ball and stick representation. You can change or add representations to the molecule. So if we wanted to change all of our protein, maybe we don't like the way this cartoon looks, we want it to all be ball and stick, we first would use a function called clear representation, and then we use a function called add representation. So let's see what that would look like. I'm going to recreate this view because if I didn't make this variable again, it would just edit this one I already have, and I want a new one. So I can do nv.showFile file path, and then I'm going to use a function called view.clear representations, and that is just going to get rid of the default representation, the default representation that NGL gives the protein. And then if I want to maybe change everything, I'll do view.add representation, and then do parentheses, and then in quotes, I'm going to type the representation that I want, and I'm going to tell it to show me ball and stick. And ball and stick, it just means that the atoms are balls and the bonds are sticks. Oop. Well, and then it didn't show me anything, so then I'm going to press enter, and then I'm going to type view as the last thing in my cell, and then it's going to show me a new view of my protein, and you'll see that the representation has changed. So now if I zoom in, you'll see that all of the atoms are balls, and the bonds are just lines between those. So I've listed a few representations that you can use here. There's cartoon, and that makes the protein look like the cartoon that we saw originally. There's also base, and that's used with nucleic acids. That will show the nucleic acid bases. It's usually used with the cartoon representation. There's also ball and stick, which is the one that I've shown here. There's licorice. It's similar to ball and stick, but it doesn't have spheres on the atoms. There's space fill, where the atoms are drawn as large spheres. And then there's also one called hyperball, which I think looks really cool. And then if you click on this link, it's going to take you to a full list of representations, and there are like a lot of them. So I only listed some of them. And then here we have a short exercise that I want everyone to, so I just want everyone to take like 2 minutes and play around with these different representations. So let's just take a few minutes and do that. And I see a question in the chat about orthographic versus perspective, and I know what you're talking about, but I meant to look it up before this workshop, and I haven't quite figured it out yet, but I am sure that there is a way to switch it. And then for someone asking how to get to the terminal, we did in the setup, if you're using Anaconda locally, we did in the setup instructions have some directions for how to install NGLView using the Anaconda Navigator. So I'm going to send those. So for the person saying it says it doesn't have clear representation function, try adding an S so it's clear representations. I think that might help. Yeah. So yeah, let me scroll up so that you can see this. It's clear representations. Okay. All right, so maybe you've had a chance to play around with a few representations. I'm going to copy this and I'm going to put it here. So I'm going to show what a few of those look like. And actually, this is a little bit of a problem. So I'm seeing that this cell, I'm not seeing my color coding here. So you'll notice here that I've got ball and stick is like kind of a red color. That's something that Python does, or the Jupyter notebook does to Python code. This is all the same color. And I'm seeing that this cell is raw in B convert. So I'm going to change that to code. Okay, that looks better. And then let me execute that. So that's ball and stick. So I'm going to look at space fill. So this is what space fill looks like. So these are just, the atoms are just large spheres. And I like this one. This one is pretty cool looking. So someone asked about how we use base. So base is only going to show things if you have bases. So if you have, so it's only going to show, you're not going to see anything on this PDB file because there's no DNA or RNA. If you choose base, it's only going to show nucleic acid bases, and there aren't any in this file, so you won't see anything. But there are some PDB files that have DNA and RNA, and you could use base in those. Yeah. All right, so that's really cool. But NGLView will also let us specifically only select certain parts of our molecule. So right now, when we're adding like a representation, it's applying it to the whole thing. And we can make it so that we're only applying a representation to like a certain part of the molecule. So if I want to only see the ligand, I can select only the ligand. So I can do view.add representation. And then I'm going to put the representation that I want, which I'm going to use this one. I think this one looks really cool. And then I can select part of the molecule by putting a comma and then typing the word ligand. And that's going to select the ligand only. There's a list of like built-in keywords that you can use. And I think I've given some of them at the end of this notebook. And then you can also see them online. So here I've added a representation, and then I have selected only the ligand, and then I can look at what I've created. And now you'll see that we're only seeing the ligand molecules. So I'm going to keep building on this. So I'm going to copy this. So we can continue to make this very fancy if we want. So we can change the color. So if I add a keyword called color and then put an equal sign, I can type the color that I'd like. So maybe I want my ligand to show up as red. And you can also change like the opacity or sort of make it see-through. I'm going to set this equal to 0.9. So just make it like a little bit see-through. And we can center our view. So I can do view.center. And then in parentheses, I'll put what I want to center my image on. And then I want to look at what I've done. So I realized that I typed all of this and then forgot to execute it because I don't see a number here. So I'm going to click inside of the cell. I'm going to execute the cell. And then let me look at this. So that is just, we've made this red, and then you'll notice that we're zoomed in on the ligand automatically. Okay, so if we want to add representations, so maybe now I want to add the protein back into my view. So this is check your understanding. So go ahead and take 2 minutes and see if you can figure out how to add the protein back, making it blue and using the cartoon representation. So we'll just pause here for 2 minutes or so. So we asked a question about the color coding. I think there was one cell where, and I think it was this one. If you aren't seeing like things in quotes as red, make sure that your cell is a code cell. So if you click inside of the cell and then select code. Okay. All right, so for this part, we wanted to add another representation. We wanted to add another representation. So I'm going to copy what I have here and just add to it. And then I'm going to copy this. So I just want them to show up on the same, I need to add my representation first, actually. So before I show it and center it, I want to add the other representation. So I'm going to do view.add representation. And then in quotes, I put the representation name that I want, which I want to do cartoon. And then I can put a comma, and then I'll type the word protein. And then I want to make it blue. And then I will do my, make it a little bit see-through with this keyword. And then I do want to center it on the ligand. You could choose whether or not you wanted to do that. And then finally, I want to see what I created. So we had a question if we can add annotation of the position of any amino acid. The answer is yes, you can. It's a little more than we are going to show in this workshop. You can add basically any label that you want or visualize any part of the atom that you want. It's depending on how complicated what you want to do is, the code might be very complicated or might be more complicated to do that. So we're doing just sort of ligand and protein, like simpler representations today. So here it is. Here's what we have when we've done this. So you'll see we have our protein here, and then we have our ligand is in red and shown in this location. I'm going to copy this part, and just continue to build on it. This is a little aside portion. You can also put in information that labels the contacts in your protein. So things like hydrogen bonds. There are ways to show specific contacts, but if you want to show all of the contacts, so I just copied what I had in the cell above, and then I'm going to add another representation. View.add representation, and then I can put in quotes contacts. And let's see, is it I think I might have missed, oh, it's contact, not contacts. So it fails silently. So now we can see all of these dotted lines. So we had a question about it's sort of like cutting the protein. I'm not sure that it is. I think that we're just getting a good angle by default where we can see this ligand really well. You'll see that here it does do this eventually. Usually in visualization programs, this has to do with some of the settings, and I haven't played with these settings in NGLView, but I imagine that they are something that you can set. Okay, so next section is saving images. So I'm going to rerun this just to see to get it how it is by default. So once you have a representation looking the way you want, you can save an image to your computer, like to maybe use in a presentation. You can embed a static image in the notebook, for example, so you can use a function called render image. So we can do view. So if we want to just make a static image in the notebook, we can do view.render image, open and close parentheses. And it will look like this at first. It will take just a minute, but I think it's creating the image, and then eventually you'll see this image appear here. So this is no longer interactive, it is just a static image. We had a question about just seeing contacts between the ligand and the protein. I think that it would be really great if there was a keyword to do that. There doesn't seem to be a built-in keyword for that. You can do it if you do a bit of scripting with the libraries. And I spent some time trying to figure it out also. But it's sort of beyond the scope, I would say, of this visual of what we're doing in this workshop. And I'll see if I can figure something out and send it out to everyone, or you all can play with it and see. It might require us to use another library, because there are other libraries that we can use NGLView in that might know more information about the ligand and protein that might help us do it. Okay, so that's rendering an image. We can also download the image. So if we want to download the image, I believe that is view.download image, open and close parentheses. And I don't know if you can see this, but now I have in my downloads folder, it's created a file called screenshot.png. If you click on that, you'll have your image. It will pop up. I don't think I shared that window, so I don't think that you can see it. But if you're following along, you can kind of see your own. And then I think this is a really cool thing. By default, it's going to be called screenshot.png. But if I want to give it a different name, I can do view.download image, and then I can put the name of the file in parentheses, in quotes. So I think this was 3fgu.png. And I might put